So far we've talked about selective breeding as one form of biotechnology that we use today. Today we're going to review selective breeding, and we're also going to move on to how we analyze genetic information from different species or organisms. So let's just recap selective breeding from the previous page of notes. In the question, you have a picture of two plants that are being brought together to create a new plant. You have the diseased African cotton plant and the healthy American cotton plant put together to produce a healthy cotton plant produced to grow in Africa. Is this, 1, this technique results in offspring that are genetically identical to the parents, 2, new varieties of organisms that can be developed by this technique known as selective breeding, 3, this technique is used by farmers to eliminate mutations in future members of the species, or 4, since the development of cloning, this technique is no longer used in agriculture. Hopefully you remember from class that selective breeding is when we choose two parents with specific traits to create desired characteristics in the offspring, such as a healthy cotton plant that's produced to grow in Africa. So we get the African gene from this plant, and we get the fact that it's healthy from this plant, put together creates a new variety. Today we're going to talk about gel electrophoresis. This is a process that we can take DNA pieces and actually separate them based on their size. So big pieces won't move far and smaller pieces will move further. Then we can compare them to different organisms to figure out how related they are. So basically how this works is we have a long strand of DNA, and we use something called restriction enzymes. Remember that enzymes have two jobs. They either cut things or put them together. In this case, restriction enzymes are used to cut the DNA at very specific locations so that every single organism that's being analyzed will be cut at the same locations if they're the same DNA. This creates DNA fragments or pieces of DNA, and then we'll show the next step, but basically they're put into something called wells right here, and then they move across this surface and stop at different locations based on their size. So that image is also in your notes, and that's really important to refer back to when we talk about gel electrophoresis. An example of a restriction enzyme is called EcoRI, which is just the strand GAATTC. So anytime this is seen in the strand of DNA, the enzyme knows to cut at that location. Keeping this the same for all of the strands of DNA makes sure that it compares every different organism or species at the same places. So let's look at this image a little closer. Step 1, the restriction enzymes cleave or break apart the DNA into smaller segments of various sizes. So we've got one long strand here, it's broken into different pieces. In this case, there are 3. Step 2, DNA segments are loaded into wells in a porous gel. So this is actually kind of a jello-like substance, and the gel floats in a buffer solution, so there's liquid actually underneath, within a chamber between two electrodes. Electrodes are here and also here. Step 3, when an electric current is passed through the chamber, the DNA fragments move toward the positively charged cathode. So the DNA gets inserted into the wells, and the DNA moves, see how there's a minus sign here, towards the plus sign. DNA has a negative charge, and remember that opposites attract, so the negative wants to move towards the positive. Step 4, smaller DNA segments move faster and farther than larger DNA segments. So if they started here, the smaller ones are going to go all the way down here, and the bigger molecules aren't going to make it very far. They're just going to stop maybe here, and then maybe the middle-sized pieces will be here. So again, here's our gel, noticing that we have a negative charge here and a positive charge on the other end. Label that on your notes right now. The DNA fragments then get added, and what are we adding here? Notice that in the picture, you see an electric current, that little lightning bolt. So we actually plug the gel electrophoresis in and allow electricity to move the DNA strands across this gel. Noticing that out of our 2 DNA strands, which one moved all the way down here, the bigger one or the smaller one? So the smaller one moved very far, almost all the way to the other electrode. However, the larger piece, notice it didn't go very far. It's much bigger, so it's going to move a little slower. Now we can see a picture of a gel after the DNA has moved across. Notice that it's been tilted a little bit. The wells that they started at were up at the top now, and that's the negative charge. Down here would be the positive charge, and the DNA would be moving down each of these wells. So noticing that there's different pieces that can be applied to each well up at the top. So this might be one organism, this could be a second, a third, a fourth, a fifth, a sixth, a seventh, and even an 8th organism, all being compared at the same time. Now when we look at these, we're looking for similarities. So we can tell already this is an oddball. So this organism, whatever it may be, is not very related to any of these. Notice that these bands are all kind of around the same area. Some are even more related than others. So for example, if we look at this row in 2 as well as row 5, we can see that they both have these 2 bands of DNA or fragments of DNA, and they also have one down here. Remembering that these are bigger and these are smaller. So let's check our understanding. DNA samples were collected from 4 children. The diagram below represents the results of a procedure that separated the DNA in each sample. The first bullet, identify the procedure used to produce the results. Can you remember what this process is called? If you're having trouble, look back at the name of the aim. That's the name of the process. Bullet number 2. The DNA is most similar in which 2 children? Support your answer. So looking at that image, figure out how similar A, B, C, and D are. Which 2 are the most similar and why? And bullet 3, state one way information obtained from this procedure can be used. Why might we want to do this procedure in the future? Also, one last question. The DNA of a human cell can be cut and rearranged by using what? A, a scalpel, B, electrophoresis, C, hormones, or D, enzymes. What do you think is the correct answer here? Hopefully you guessed D, enzymes. Remember that restriction enzymes are used to cut the DNA.